Hey everyone, thank you so much for tuning in. My name is Shanta Hejmadi. I use she, her pronouns, and I'm a PhD candidate in ecology, evolution, and behavior at the University of Minnesota. And before I begin, I want to acknowledge that we're all sitting on stolen land. Our home's Dakota name is Minnesota Makoche, and its Ojibwe name is Manazadi Waking. I also want to acknowledge that all of us are sitting very near the place where George Floyd was murdered, some closer, some farther. And you might be wondering why I bring these things up at a scientific paper session. But the fact is that our colonial history of genocide and white supremacy has shaped more than just whose ancestral land we own, who has been able to buy houses, and who is more likely to be killed by police. That history has also shaped who feels welcome in birdwatching circles, as we saw during Black Birders Week this year. It has shaped our science and our scientists in ways that exclude Black, Brown, and Indigenous voices. And I ask that each of you consider how these systemic forces have shaped you and how you can fight them. So I know that I gave you a very academic and somewhat incomprehensible title here for this talk, but let's fix that right now. I would like to invite you on a journey around the world with me in a talk that I would really rather call Fantastic Raptors and Where to Find Them. I'm going to share with you some preliminary results of my dissertation research on global communities of diurnal birds of prey. I figure since none of us can travel because of COVID, we may as well travel together with science. So we all love birds, right? That's why we're here. But more generally, we're all interested in biodiversity, where it came from, how it's maintained. And there is lots of ways to study this. For instance, we might look at how many species occur in a particular place, or species richness, like on this heat map, where red indicates greater numbers of species and blue indicates fewer. We might ask how things are related to each other with phylogenetic trees like this one, also known as an evolutionary tree or a phylogeny. Or we might ask questions about morphological or functional diversity. For instance, you can see how different these bird beaks are, and you can bet that their owners do really different things with them. So I'm a student in ecology, evolution, and behavior. And those first two fields, ecology and evolution, have really different ways of looking at biodiversity. But integrating the two approaches can lead to new and exciting insights that begin to get at this big question of how can we understand the origins and maintenance of biodiversity on a global scale. My research sits at this intersection of ecology and evolution, and the question I'm going to address today is, can data about how organisms are related to their neighbors, how similar they are to their neighbors, where they come from, and the history of the land itself give insight into how their communities assembled? And I'm going to address this question about global communities using diurnal raptors, which fall into these two groups, Accipitriformes and Falconiformes. Get ready for some cool bird pictures. So these two groups of mostly carnivorous birds are a really great system to ask global questions about community assembly. There's about 333 species. Both of the groups are globally distributed, and yet we have two groups here that have to go back 65 million years to find a common ancestor. And yet, despite that, they have really striking morphological similarities. So Accipitriformes is your hawks and eagles and vultures, and Falconiformes is your falcons, falconets, and caracaras, but the layperson might not know which is which because they're so similar. Probably folks that are listening to this would because you all are bird watchers, but the average person might not. There's 267 species of Accipitriformes. This group encompasses your hawks, eagles, vultures, and kites. Sometimes you'll hear me just call them hawks. I'm not trying to say anything bad about eagles, vultures, and kites. I'm just saying hawks to encompass everybody for time's sake. There are 67 falconiforms, which include falcons, forest falcons, these adorable little falconets down in the corner there, as well as caracaras. And sometimes you'll hear me call them all falcons. So as you can guess, Within both of these groups, there are families and subfamilies. I'm not really going to get into that, but I got to tell you something bonkers. You know how I said that hawks and falcons have to go back 65 million years to find a common ancestor? That means that all the birds on this slide are more closely related to the northern cardinal in your backyard than they are to any hawk, eagle, or vulture. And I said I was going to study how communities assembled. Well, what do I mean by that? Asking how communities assembled is just one way of interrogating big questions about biodiversity. And to illustrate, here I have two raptor communities from opposite sides of the world, one from the Amazon basin in South America and one from a southeastern coastal forest of Australia. Now I'm going to superimpose another image with these species, with these communities. These are phylogenies of these species. They're evolutionary trees. They're meant to be read from the center outward, and they illustrate the relationships between the species. But what I really want you to pay attention to here are the green and black tips around the outsides. Green tips indicate species present in the community, and black tips indicate species not present in the community. And you can see that in the Amazon basin, there seems like there's more clumps of species that are all from similar parts of the tree than there are in the southeastern coast of Australia, which seems to be a little more spread out. But let's put a few numbers on this, shall we? In South America here, we have both groups, Falconiformes and Accipitriformes, your hawks and your falcons. We have 3 families, 23 genera, 33 species, 5 cathardid vultures, those are relatives of our turkey vulture, 19 other hawks and eagles, and 9 falcon species from 3 distinct groups. Whereas over in southeastern Australia, while we still have both orders represented, we have only 2 families, 11 genera, 19 species, no vultures at all, 14 other hawks and eagles, and 5 falcon species from a single group, in fact, from a single genus. My research asks, how did this happen? Why are these species here and those species there? How did they get there? And how is it maintained through time? And telling a rich story of how it happened, especially over the whole world, involves, for me, asking how closely related community members are, how physically similar they are, when and how they got there, and how the history of the geography itself influenced who ended up there. So what did I do? Well, to start with, I constructed a biogeographic history, which is basically an analysis that uses an evolutionary tree or phylogeny and species distributions to infer when certain groups of critters arrived in a particular geographic region. Then we ask, how closely related are the species in a community, and how physically similar are they? So I divided the whole world up into 1 degree by 1 degree grid cells, defining that community as that grid cell. And I used the phylogeny as well as a whole lot of measurements that I took off of bird specimens in museums to calculate how closely related and how similar species in a community are. Multiply that by 15,000 communities, and I can compare patterns across the whole world to see what it tells me. And before I get into my results, I've got to digress for just a minute and give a shout out to the museums where I've been collecting data, as well as my undergraduate helpers. I love museums, and I love mentoring undergrads. These folks in the picture that are not me here are Lydia, Michaela, and Andrew, and these are just 3 of my undergrads. There's 6 others that are not pictured, but in the summer of 2018, these 3 went with me to the American Museum in New York City and the Field Museum in Chicago, and they helped me collect morphological data on over 1,100 specimens. And this also brings me to a really important topic, which is the history of museum collections. These collections are amazing sources of information on the world's biodiversity. I mean, it's pretty incredible when you think about the fact that I am learning new things about diurnal raptors using specimens that are often over 100 years old. But something that I'm always conscious of in these collections is the legacy of colonialism, racism, and oppression that goes with them. Many specimens were collected with the assistance and sometimes exploitation of local, indigenous, and even enslaved naturalists. And this becomes more true the further back in the past you go. These folks who contributed their knowledge then get no credit. They got no lasting benefit to themselves or their community. They got no access to the specimens, which are stored under high security, often thousands of miles away, and in countries with restrictive visa laws. I'm probably never going to get to learn the names and histories of the Black, Indigenous, and people of color who contributed to collecting the specimens I've used in my work. So for the moment, John Edmonstone, who was the former slave that taught Charles Darwin how to preserve animal specimens, will have to stand in for them for now. But while museum collections do have this fraught history, they are incredible treasure troves of information. I love them, and my research relies on them heavily. And I want to talk briefly about what it looks like to collect morphological data on these specimens. Basically, I take a specimen and I measure the wings and the feet and the tail and beak, including the wing cord, the secondary flight feathers, the tail. I also measure toe length, as well as the length and width of claws, and I measure the length and width of the bill. And these measurements on specimens comprise what we call morphological data, or if we want to use simple language, traits. But you can see how these measurements, especially in the case of the bill, don't capture a lot of the information contained in the whole shape. I mean, this is a cool looking beak, and I'm reducing it to only 2 linear measurements. And this is why I also took over 631 scans of beaks, in addition to the over 16,000 linear measurements on 1,107 specimens of 309 species. I am actually still in the midst of processing these beak scans, so unfortunately I can't share them with you today, other than these cool pictures. But I can share with you the linear measurements that I took. This figure summarizes the shape variation in the over 16,000 linear measurements, with hawks, eagles, and vultures are accipitriforms in yellow, and forest falcons, caracaras, and falconets in purple are falconiformes. These axes on this plot represent different dimensions of variation in shape as defined by those trait data. And these data are interesting just by themselves, but unfortunately, I don't have time to say really anything more about them today. But suffice it to say that these are the trait data that will be represented in the analyses that I show you today. So remember when I said I was calculating how closely related species are and how physically similar they are? Well, compared to what? How do we know if they're more closely related than we would expect? Well, let's say, for instance, that we have 3 communities here. What we do is we use a computer program to put all of these species into a hat and randomly draw sets of species. If we do that 1,000 or 10,000 times, we end up with a distribution of pretend communities that has a range of values that we expect to see most communities fall into. This is our null distribution. So let's say community 1, for instance, has really distantly related species. I mean, that rubber duck is messing things up. And if we were to plot this on the expected distribution, it will show up outside the expected range in this distantly related part of the plot. Community 2 has a lot of closely related species. They're all chickens and ducks. And so when we plot that one, it's also going to be outside the expected range, but on the closely related end of things. Community 3 is kind of a random mishmash. So when we plot that one, it's going to be right inside that expected range. And when relatedness of a community falls outside that expected range, we say it is significantly different than our null expectation. And the same is true of trait diversity that we see in these communities. In this case, our null distribution will have an expected amount of trait diversity. And so things are on a scale from less diversity to more diversity instead of closely related to distantly related. I didn't change the plot here, but trait diversity and relatedness do not necessarily have to have the same pattern. However, it is still true that when the observed community data falls outside the expected range, we say it's significantly different than expected. So, are there communities where relatedness and trait diversity are significantly different from the null models? And the answer is yes. Here I have relatedness on the left and trait diversity on the right. Dark blue means species in that community are more similar than expected, and bright yellow means species in that community are more different than expected. But to really learn anything meaningful about the relationships between trait diversity and relatedness, we need the whole continuum of these values that I'm showing you now. So these are the first global analyses to include both relatedness and trait diversity in all diurnal raptors, at least that I'm aware of. And remember, I promised you a journey around the world. Well, we're going to look more closely at Africa, Australia, and South America. So remember, we want to know about the relationship between these two variables, between relatedness and trait diversity. So we need a slightly more sophisticated null expectation than what we just talked about. What it's going to look like when we're thinking about relatedness and trait diversity on an entire continent. So let's imagine this two-dimensional density plot that I just put up with relatedness on the x-axis and trait diversity on the y-axis. When we plot a community in this space, its position tells us where it falls on the scales of relatedness and trait diversity. Communities that fall outside these yellow lines are significantly different from the null expectation on the relevant axis. So this one is significantly different on the relatedness axis, but not on the trait diversity axis. As we add communities to this plot, the denser parts of the space where there are more communities will become brighter. And we don't actually have any examples of what this distribution should look like for a global group of organisms. So I need a null expectation to compare it to when I plot my real data. My first pass at a null model asks, is what we see different from random assembly? And I'll show you how it works with Africa as an example. What we do is we shuffle the species in every community in Africa and recalculate the trait diversity and relatedness for this random or null community. We do it 100 times, and then the maps of the null expectation for relatedness and trait diversity look like this, basically a uniform blue-gray right in the middle of the scale. All of the continents look like this. And when I plot these data from the null model, the density plot looks like this. You can think of this like a topographical map with contour lines where tighter contour lines and brighter colors mean more communities in that part of the variable space. So how does this compare with what I observed in the relationship between relatedness and trait diversity in Africa? Well, looking at the geographic heat maps for dispersion, you can see that there is obviously more structure than the null model. We can see that Madagascar is off doing its own thing, where it has fairly low trait diversity, but relatively more distant relatedness. And we can see that Congo has both closely related and relatively similar species. And this structure translates to structure in the relationship between relatedness and trait diversity, which is significantly different from our null expectation. We can even see the geographic signature on this purple plot, with Madagascar contributing heavily to this bright area and with the Congo contributing heavily to this bright area. Now, my biogeographic history, remember, that's the inference of when species arrived in a particular region, indicates that hawks originally evolved in Africa and then spread to other parts of the world. And the popular story of Africa's last 55 million years is this long, steady transition from mostly tropical forests to mostly woodlands, grasslands, and deserts. To me, this suggests that species originated and evolved on this continent, species of hawks, and that they had a steady biogeographic spread over time on the continent as a whole. But my caveat is that these results are preliminary, and we can see just from these maps that Africa is not a monolith, and there's a lot more geographic variation to explore here. In Australasia, which includes Australia, New Zealand, Papua New Guinea, and parts of Indonesia, the null expectation looks about the same. However, we see a totally different pattern on the map. It's more homogeneous, but it's opposite. And I also want to point out that something a little bit different is going on here on these islands, but the continent of Australia is so much bigger that it's basically going to swamp out any signal that we might see from that. So here's this pattern that we see in Australia, where it's almost homogenously yellow and almost homogenously blue. on our two different variables. And this pattern is even clearer when you plot the observed data. In case you're not sure, yes, this is significantly different from the null model. Communities in Australasia are distantly related and have relatively less trait diversity. Now, my biogeography indicates that there were colonization events into Australia as far back as 17 million years ago, but most of them have actually happened in the last 6 million years. We also know that Australian woodlands and grasslands began expanding around 16 million years ago, and that the most recent 2.5 million years have been marked by rapid forest contraction, grassland expansion, and aridification. So to my mind, this is consistent with a scenario of widespread extinction due to rapid landscape change as those forests retreated to the eastern coast, followed by this ecological opportunity in this newly harsh environment that allowed for the colonization of distantly related birds that were already adapted to be successful there. And finally, South America. For this continent, I separated the hawks from the falcons because there were so many falcons. So the null models for Accipitriformes look like all the other ones, but the map clearly shows us there's going to be more structure. Species are relatively distantly related, and there's a lot of variation in how similar they are in terms of trait diversity. And I don't have time to point out all the geographic features here, but one thing that is really interesting here to me is the signature that you can see of the Amazon River, and I'll take it out and put it back again so that you can see that really well. And sure enough, the density of this distribution of hawks is significantly different from the null model. It's pretty tightly concentrated over in distantly related territory, but it's much more spread out in terms of what we see for trait diversity. My biogeography indicated that a cathardid vulture ancestor colonized around 30 million years ago. Those are relatives of our turkey vulture, and it then diversified into more species. And there was also a Bateo-like hawk ancestor that colonized around 9 million years ago and diversified into a whole bunch of species, as well as we've got about 12 other fairly recent colonizations. So in this case, we have both old and new events where an ancestor colonized and then diversified into more species. And this results in these relatively distant relatedness metrics, as well as this broad range of trait diversity in communities. But we can't complete the story of South America without falconiformes, of course. The null distribution for falcons looks like all the other ones, but the map tells us there's going to be some structure here, right? We can clearly see the Amazon has lower trait diversity, and we can really clearly see that these southern arid grasslands have much lower relatedness. Or sorry, much closer relatedness, excuse me. Now, when we plot this density plot, the structure is clearly there. And similar to what we saw in Africa, the Amazon is contributing heavily to this bright spot, and the southern grasslands are contributing heavily to this bright spot. Now, I am not the first person to do a biogeography of falcons, but my analysis confirms what we already knew, which is that falconiforms originally evolved in South America. In fact, 80% of falconiform species here are from two groups, the forest falcons and the caracaras, with only very few of the falcons that are close relatives of the peregrine falcons that we see around here. So it's been suggested that the diversification of these groups has been driven both by grassland expansion that began in the early to mid-Miocene period, something like 15-ish million years ago, as well as rapid expansions and contractions of various habitats across the continent that ensued all the way up through recent times. So in South America, we've got some instances where species originally evolved on the continent, with speciation potentially being driven by rapidly changing habitats. We've got some cases where species evolved elsewhere and colonized South America only to continue generating new species after they got there. And we've also got a few cases of recent colonizations. And these processes are all happening at multiple points all throughout the last 30 million years. In other words, there is a lot more to do here. And raptor community assembly in South America is driven by processes whose diversity potentially rivals the biodiversity on the continent itself. So clearly I have a lot more to do. I am in my 4th year of my PhD right now. First off, the phylogenetic tree that I'm using is a good start, but it has a lot of flaws and I need to make my own tree, which is more comprehensive. With that new tree, I can do a new biogeography, which will have better dates on it. And we're also currently working on more sophisticated null models that are actually going to account for evolution as they're generating those null expectations. And lastly, I still have a whole bunch of those three-dimensional scans that I need to process and add to these analyses. So just to come full circle, and this question of how do we understand the origins and maintenance of biodiversity, the results I've shared with you today are preliminary, but they're still pretty exciting. I'm excited about them. We've got 3 continents with 3 distinct stories of community assembly and diurnal raptors. I've documented global patterns of relatedness and trait diversity in raptor communities for the first time that I'm aware of. And finally, this work has resulted in one of the first examples of how millions of years of community assembly has unfolded on a global scale. With that, I want to acknowledge a lot of people and funders, from my lab group to my undergrads to my committee and my wonderful family support network. I would also be remiss if I didn't acknowledge the staff at the American and Field Museums and how heavily my work relies on museum collections. Collections which, as I touched on briefly, are treasure troves of data, but are fraught with these issues of colonial era collecting on indigenous lands. And this is particularly true of the specimens that I study, which tend to be very old. This difficult work of decolonizing museums while still letting science happen is difficult but critical work, and I'm committed to doing my part as a Bell Museum graduate student. And I look forward to a day when museums and museum-based science and all bird science and bird watching is accessible, inclusive, safe, and welcoming for everyone. And with that, I hope that you'll join us at the roundtable discussion on Saturday. I would love to take any questions that you have at that time. And if you can't make it, here's my email address. Feel free to email me anytime. I would love to hear from you.